Hello, my name is Dr. Roderick L. Roll, and today we will be discussing Mendel and the gene idea. Gregor Mendel was a monk who studied garden peas, and from his observations, he is named the father of genetics. He analyzed patterns of inheritance in these garden peas. Why did Gregor Mendel choose the pea? Well, you have to look at the anatomy of the flower, which is the reproductive part of the pea plant. So pea plant is a plant that will produce a flower. We call it the flowering plants. It has a male portion and a female portion. The male portion is referred to as the stamen, and the female portion is called the pistil. The sperm will be produced in the stamen, and the egg will be produced in the pistil. While growing these garden peas at the monastery, Gregor Mendel made three key decisions. The first is that he used what is called a true breeding plant. Secondly, he controlled breeding. Thirdly, he observed 7 different characteristics or traits. We will discuss each of these. The first one we will discuss is the use of the true breeding plants. So what is a true breeding plant? If a plant is only mated with itself, it will only produce offspring that look identical. So in the image we have a purple pea plant and a white flowered pea plant. So every time that white flower pea plant produces babies in the form of a peas in a pea pod, those peas will develop into another white flowered pea plant, and vice versa with the purple. So how did Mendel control the breeding? Was he used the true breeding plants. These true breeding plants will always pass down the certain characteristic that he was looking for. Self-fertilization is what plants can do. They can also cross-pollinate. So self-pollination does occur naturally, but it's much more rare than cross-pollination. Self-fertilization is when that purple plant fertilizes itself, or the white flowered plant fertilizes itself. Typically, the purple flowered plant will produce sperm that will leave that plant and land on the stigma of the female white flowered plant. Therefore, this is referred to as cross-pollination. Self-pollination creates a single variety or a single type. The type can be purple flowered plant or a white flowered plant. So a purple flowered plant will fertilize itself by releasing sperm. The sperm will then land on its own stigma. The pollen tube will then grow through mitotic divisions down the style of the pistil. Once the pollen tube has reached the egg, sperm nuclei will fertilize that egg, therefore turning it into a pea. This pea is the embryo, and it can develop, if planted, into a new plant. Another key decision that Mendel chose is to take control of breeding. So Mendel cut off the stamen. The stamen is the structure that produces the sperm. If you cut off the stamen, then you can control where the sperm lands. So if you had two rooms, one with white flowered peas, the other room with purple flowered peas, you can manipulate fertilization. If you pull the stamen off of a purple flower, then that sperm will produce another purple flower if it fertilizes an egg. So Mendel manipulated and he controlled breeding. And because of that, he was able to look at these crosses and to create the crosses. So Mendel controlled breeding by crossing true breeding plants, which produced an F1 generation. He interrupted the self-pollination process by removing the male flower part and exchanging the pollen himself. This is the control of breeding. This produced the desired characteristics that he wanted. The third key decision that Mendel chose is to look at multiple characteristics, to look at multiple traits. Mendel chose 7 traits. Some of them are, he looked at the pod shape, he looked at the pod color, he looked at the stem length. As you look at these different traits, you notice that whenever he crossed a true breed with a true breed, he always got a certain ratio in reference to the F2 progeny. All of the F2 progeny gave him a 3-to-1 ratio. And all of this was obtained because Mendel manipulated the pea plant. He, 1, created the true breeding plants. 2, took control over breeding. Number 3, used several traits or characteristics. These are the other traits to make the 7: seed color and seed shape. This is the offspring of a fertilization cross. You will produce a pea pod that has individual peas inside. Each pea is an embryo. From the crosses that Mendel observed, he created the concept of genetics. So we're going to go over some of the terms now that were eventually created because of his crosses. The first one is a monohybrid cross. A monohybrid cross is a cross that looks at only one trait or only one characteristic. This characteristic or trait is a product of the gene itself. So one example is flower color, purple flower versus white flower. A character is a heritable feature that varies among individuals. Again, flower color is the example we'll use. Some flowers were purple, some flowers were white. Each variant of the character is called a trait. Each variation is called a trait. You have the purple flower versus the white flower. These are traits. It is produced by one gene, and that gene is located at one position on the chromosome. That one position is referred to as a locus or a location. So if one variation overshadows another in the F1 results, then the inheritance pattern is called complete dominance. So in Mendel's original cross of one of these traits, purple flower versus white flower, when Mendel crossed those two, in his results, in his F1 results, Mendel saw that all of the offspring were purple. This meant that one allele, one character, overshadowed the other character. This means that one trait overshadowed the other trait. Modern terminology versus old terminology. A gene is a DNA sequence for a heritable feature. So heritable feature was a Mendel term, the character. Now that we have sequencers, we know that there's a sequence of nucleotides that code for the character. The next one is alleles. Alleles are different DNA sequences for the same gene. Mendel used a variant, a variation, a variant form, an alternative form. Now we use the term alleles. So a trait is what you physically see. The trait is what you physically see. The purple flower versus the white flower. So now we're going to look at some examples of Mendelian monohybrid crosses. You start off with true breeding plants. You have a purple flower versus a white flower. We're going to call those the parental types or the P generation. Then we're going to cross those two and create the offspring. The offspring or the babies will be called the F1 generation, the filial. So the filial or the babies, they would all be purple. Now, if we take two babies and cross them with themselves, you produce an F2 progeny or an F2 generation. When Mendel performed this cross, he saw that the white flowered plant that disappeared in the first cross reappeared. And it reappeared at a 25% ratio or 1/4 ratio. So the raw data collected from Mendel's crosses illustrated that every one of his 7 characteristics produced a 3-to-1 ratio when in reference to the F2 progeny. Let's look at another example. What type of gametes will be produced if the parent plant has a genotype of big P, big P? So that was one of the examples of the true breeding plant. Purple flower is big P, big P. If you take that flower, you take one of the cells and take it through meiosis. What I'm asking is, the gamete that will be produced, that one daughter cell, what will it have in it in reference to P's? It will only have a big P. If we looked at the white flower, it originally had 2 little p's. If you take that through meiosis 1 and 2, and you looked at the gamete, you would only produce a little p. So that's the question that I'm asking. I tried to draw it to help you understand it better. If you have big P and a big P, they are genes located on a chromosome at a specific region. That cell is going to go through interphase, and the genetic material is going to be duplicated. Now you have this tetrad. You're going to separate the tetrad in anaphase 1. Now you have dyads in each chromosome. In each daughter cell. Then you'll take those daughter cells through meiosis 2. And now you're going to end up with 4 daughter cells. Each daughter cell will only have 1 big P. Because remember, from meiosis, you're going to half the chromosome number. If I ask you on a test that question, what will be the gamete that will be formed from big P, your answer would be just 1 big P. Here's the second question. What type of gametes will be produced if the parent plant has the genotype of little p? Same concept. This is another true breeding plant. If you take this through meiosis 1 and 2, you can only produce a little p. I also drew this one. If you have little p in the beginning, replicate everything in S phase of interphase, and then take this cell through meiosis 1 and 2 to produce 4 daughter cells. Each daughter cell will only have half of the original genetic content. If I asked you on the test to give me the gamete that would be produced from that original parent, you would put a little p. Now, whenever you're using a lowercase letter that looks just like the uppercase letter, you need to put a overscore, a little line over the little p. Here's another question. What type of gametes will be produced if the parent plant has the genotype of big P, little p? Same concept. Draw this out through meiosis 1 and meiosis 2, and half of the daughter cells would have a big P in it, and half of the daughter cells would have a little p in it. So you can put the big P and circle it, or you can just put the big P. You can put the little p and circle it, or you just need to put a little p with an overscore over it. So we're looking at the heterozygote. We're looking at the offspring from the original cross of true breeding plants. It has a big P and a little p. So 50% of the time you'll get a big P, and 50% of the time you'll get a little p. If you were writing this out, you would write big P or little p. The next question comes from your lab manual. Are the chromosomes in the new cells different or identical to the ones in the original cell? To help you with this question, I drew it out. Originally, we have one long chromosome that's black and one long chromosome that's red. The black is a big P and the red is a little p. During meiosis, this cell is going to replicate its genetic material. A tetrad is going to form in prophase 1. Remember, we have 5 stages of prophase 1 where crossing over will take place. This means that the 2 chromatids that are closest together, that are non-sisters, they are going to have breakage and then cohesion of new portions or new parts. Therefore, the 2 daughter cells at the bottom that are on the outsides of the drawing are all original, whereas the 2 daughter cells that are in the middle have recombined. Their DNA has broken off and migrated to another chromosome and reannealed, therefore creating recombined types. So the answer to the question is 50% will be the same and 50% will be different. Here's another question. Are there any new genetic material in any of the daughter cells? The answer is no. There are no new genetic material. Why? If there was new genetic material, then the genetic material would come from outside of the cell. So no, you only have recombined DNA. I tried to draw what new DNA would look like. In the bottom image, we have recombined DNA versus a recombined chromosome. Recombined chromosome is some of our genetic material from one chromosome, let's say from the parental copy, gets cut off and moves to the maternal copy. Versus the one on the bottom, that is saying that foreign DNA is going to come in and be united with the original DNA. Here's another question. What are the gametes produced from the following P generation genotypes? So if we have big A, what we're asking is, if you take that through meiosis 1 and 2, and we look at one of the daughter cells, what will be in a daughter cell? And the answer is just one big A. So you should be able to take the rest of these and figure out the answers to them. Now we're going to answer another question. This is the opposite of what you just looked at. What is the genotype that produced the following gamete? So the first one we'll look at is if you have one gamete or one daughter cell that has a big A in it and one daughter cell that has a little a in it. What we're asking is, who's the mom or who's the dad, or what created that gamete? The answer is a father or a mother that has the genotype of big A and a little a. Use the multiplication rule to determine the offspring probability. The multiplication rule is just going to utilize pennies. We're going to flip coins. And flipping the coin is equivalent to having 2 types of variations. When we just said the purple flower versus the white flower, that was a big P versus a little p. So you only have 2 variations that you can have. So we're going to use the multiplication rule to determine the offspring probability. So in a heterozygote individual, each gamete has a 50% chance of carrying the dominant allele and a 50% chance of carrying the recessive allele. Look at the arrows. So for the sperm that comes from dad, he has a 50% chance of giving a big R, and he has a 50% chance of giving a little r. This is if the dad is a heterozygote. If mom is also a heterozygote, she has a 50% chance also. So you would put her on the far left side where it has the egg, and 50% of the chance for her to give a big R and 50% chance for her to give a little r. So let's look at how to use this multiplication rule. The probability that two or more independent events will occur together in some specific combination can be determined using this rule. So we're looking at that first square. So we have a big square in the bottom. Let's look at the first square where those two red arrows are pointing to. So we're looking at that individual. So we're going to use the multiplication rule. So we're going to look at event 1 is a 1/2 chance to get a big R. Event 2 is a 1/2 chance to get a big R. So event 1 and event 2 is going to produce that individual that's in that box. If we look at a different box, we'll have to use event 3 and event 4 and 5, 6, and so on. So let's just look at that first box. So there is a 1/2 chance to get big R, event 1, and a 1/2 chance to get a big R, event 2. You multiply those two together using the multiplication rule, and you're going to get a 1/4 chance that you'll get a big R and a big R. So therefore, that's how we've determined what the probability of receiving a big R and a big R as an offspring if you had a heterozygote crossed with a heterozygote. A better way to do this is to just draw a Punnett square. So the square is easy to draw. So we're going to look at how to draw this. So you would draw the square, and you'll go back to the parent. One parent is going to produce the sperm, and one parent is going to produce the egg. And you have to figure out what is the genotype of each parent. Once you figured out the genotype, you would then decipher what gametes will be produced. If they were both heterozygote, then that would mean 1/2 chance to produce a big R, 1/2 chance to produce a little r. So you put your fractions there, and then now you can see how easy it is to determine the fractions for the boxes that are inside. So the chance that you get a homozygous dominant individual, big R, is 1/4. What is the probability to get a big R and a little r? You're going to have 2/4 because you have two 1/4. Then the probability to get a little r is 1/4. How do we determine the probability of obtaining a heterozygote if two heterozygotes are crossed? So to answer that question, we're going to use the additive rule. So let's look at the actual cross below. We have a heterozygous individual crossed with a heterozygous individual. Each of them have the genotype of big R, little r. What the question asked is if you cross two people that had the genotype of big R, little r, and big R, little r, what would be the progeny? How many of them would be heterozygote? So in the image, I circle the two that would be heterozygote. So that is the question. So you could do the cross to obtain the answer, or you can use the additive rule. How do you use the additive rule? There's a 1/4 chance that you'll get a big R, little r, or to get a heterozygous individual. There's also another heterozygote individual in this cross. So you just add them together. 1/4 plus 1/4 equals 2/4. Again, you can draw the Punnett square and physically see it, or you can just mathematically calculate it by adding 1/4 plus 1/4. I would like you to now perform a Punnett square on this cross. Please draw out the Punnett square. Take a purple flower and cross it with a white flower. If you are not finished, please pause the video. This is how it should look. Big P's are the ones to the far left. The little p's are the ones on the top. You're going to multiply this or bring this down and across. You're going to do the next ones the same way. And then now you're done with the Punnett square. It's that easy. The cross between big P, and an individual with little p, is going to yield all offspring that are going to look the same. They're all going to be big P, little p. They're all going to be heterozygotes. If I ask you for the genotype, I'm expecting you to give me the letters. If I ask you for the phenotype, I need you to give me what it looks like. Is it going to be purple or is it going to be white? So that is going to be the physical characteristics, what you see. The genotype is the genetic makeup. So technically you're supposed to write out the sequence. But instead of writing out the sequence, we have given the sequences letters. The sequence that is dominant, we give it a dominant letter. If it's purple versus white and the purple is dominant, they typically pick the letter P. So again, the genetic sequence is technically the genotype, but just use the letters that were denoted for it to represent that sequence. The phenotype is the physical characteristic, what you see, what you taste, what you feel, something that can be detected with your senses. So this is how the results should look from two F1 progeny being crossed, or two siblings being crossed would give more siblings, but they would be an F2 progeny. So the F2 progeny is what we're looking at. If I asked you for the phenotypic ratio, it should be 3 purple versus 1 white. If I ask you for the genotypic ratio, it will be 1 big P, 2 big P, little p's, and 1 little p. If I ask you for just the ratios, then you would say 3:1 or 1:2:1. Perform a cross between this F1 generation. The F1 generation in question is in the image. It is purple. So you have to figure out what is the genotype. I gave you the phenotype. If you're not done, please push pause. Two heterozygotes crossed will look similar to this. So therefore you would have one big P, two big P little p's, and one little p little p. F1 cross or heterozygous cross with another heterozygous will yield the following results. 1/4 big P, 2/4 big P, 1/4 little p. That is the genotypic ratio. The phenotypic ratio is 3/4 purple, 1/4 white. This is the cross, starting with the parental to create the F1, then crossing the F1 to produce the F2. This is another way to look at the results.